So can I share my screen now? You can let me know if.
Yeah, we have you fine. We can see and hear you fine. Yeah, perfect.
Awesome, awesome.
Perfect.
So maybe I can. Yep. So I'm going to talk today about how graph neural networks have been used for drug combinations. But first I'll give a high-level overview about how machine learning has been transforming drug development. So we've seen recent advances in molecule generation, so using data sets of molecules to generate new molecules, and molecule property prediction, which is also a supervised learning task. And there have been also how to predict how to produce these molecules, so through retrosynthesis and so on. But today I'm going to focus on adverse side effects. And I'll give a short introduction about why this problem is very important. So prescription drug use has been increased by 8% during these 10 years. But more importantly, the number of people who are taking more than 5 drugs at the same time has been also increasing quite steadily. And we can see that in the last only 15 years, how much we spent per drugs is doubled. So what is polypharmacy? And so in order to treat complex or chronic diseases, or even when we have multiple conditions, we might have to take multiple drugs at the same time. And this is what we call polypharmacy. So it's the concurrent use of multiple medications by a patient. And sometimes when these drugs who we regard as the good guys are acting in the same environment, they might end up doing some damage around. So these are the side effects we're going to look at. And side effects have been affecting 15% of the population in the US. These numbers are for the US. And treatment costs are exceeding 177 billions per year. And the problem is that these side effects in the whole drug development pipeline, testing pipeline, they are caught towards the end of the testing pipeline, so in phase 4 of clinical trials. And while some of them are discovered in this phase, most of them, or a lot of them, are undiscovered when the drugs are put on the market. And this is especially like this in the case of polypharmacy, where testing for all combinations is very expensive and requires a lot of resources. So just to give an overview of how the drug development pipeline looks like. So we have the drug discovery, which is this green square here, in which we are interested in finding the protein of interest. So which protein the drug should target in order to be efficient and cure the disease in a way. And then it's also concerning itself with molecule fabrication. So how do we find the drug and how do we produce it? And then after a formula is discovered, then there's the preclinical research, where this is done in the lab. So there are different ways of testing that the drug is not safe and it doesn't have obvious side effects. And then after the preclinical research has been done, there's the clinical research, and this has multiple stages. For each stage, there's more and more people, more and more setups in which it's tested. And after it's been tested through these 3 phases, there's the drug review and approval part. So this is the filing, everything that needs to be filed, getting it approved. And if it gets approved, which this whole process tends to last somewhere around 10-15 years. And after that, there's phase 4. So what this means is that after it's been put on the market, the doctors and people generally report if it has any side effects in the wild. And even after it's put on the market, the drug is being monitored. So that's what this phase 4 means. And this is exactly where the drug-drug interaction, so the side effects coming from taking multiple drugs, is caught in a way. So this is where people report it. This is where these things are discovered. And there's no way to test for all adverse interactions until after, because it would require a lot of combinations basically. So it's n squared in the number of drugs on the market. But in this case, machine learning can help. So we can design this as a supervised learning task. So we can take existing data sets of drug pairs, and we can either do a binary prediction. So by looking at two drugs, drug 1 and drug 2 here, we can say, and the side effects, so tachycardia, we can say, will the side effect happen or not? And the other alternative is to do, to try to predict all the labels at the same time. So for a pair of drugs, which side effects can happen? So there's basically the multi-label setup. And there have been a lot of recent work trying to model, so basically to predict side effects coming from drug-drug interactions. And one approach is to build a graph, so a big network of proteins and drugs, and which proteins are targeted by which drugs, and which drugs, drug-drug interactions are known, and try to predict the missing links. So basically, drug-drug links we don't know of, those could be side effects. But instead, our work treats the molecular structure. So we look in a way at in a finer grain. We look at the molecular structure of the drugs in this network. And so, okay, now we know we're going to look at the drug structure, the chemical structure, but how do we do this? So previously, molecules have been represented as strings using this interesting way of representing called smiles. But representing it in this way means discarding valuable information. So, and it also means that we have to, the machine learning model has to learn the rules associated with this grammar in a way when parsing it. But here we're going to represent the input drugs as graphs where atoms are nodes, so and bonds are edges. And well, graphs are everywhere really. So as we've been talking, the protein-protein interaction network is a graph. The molecule is a graph, but then there's also roadmaps and knowledge graphs. All of these things are a graph. And machine learning has been for a long time looking specifically at image structured data, so for which we have convolutional neural networks, or for textual information for which recurrent neural networks have been very useful. But for graph neural networks, it's been only more recent that we've had a specialized architecture. So I'm going to explain a bit how the graph convolutional model works. And we can do this by first looking at the convolutional neural network we know from images. So here is the general cat picture, and I'm showing in a way like what a convolutional operation does for images. So we have this kernel which is applied over the image and results in this later matrix. And in a similar way, the graph convolution can be seen by one neighbor using the information from the neighboring nodes. In the previous case, it would have been the neighboring pixels to update the information of the node. So this is one way to look at graph convolution. And in our case, we devised a novel way of using this graph convolution by doing so. As I was saying, we're looking at drug pairs, and here we're going to do a graph convolution across drugs, so between drugs in a way. So we're going to let atoms in a drug, so nodes, attend over nodes in the other drug. So it would be like one pixel in an image, look at other pixels in a second image. So this is the CrossDrag Attention. And this is what we call Coattention, and the final architecture uses the message passing, which is an umbrella term for the graph convolution, the Coattention, which is the one between drugs, and updates each individual drug, so each individual atom in the drug representation using this information. And in the end, after we update the nodes in each graph, then we aggregate this node information into one final drug level information. So for each drug, we'll get one embedding. This is denoted here as the readout dx. So for a pair dx dy, we'll get two embeddings. And then by looking at these two embeddings together, we'll get the prediction, which is in our case, whether one side effect will happen or not, or which side effect will happen in particular. There's just a short summary of what we've done. So this way of looking at chemical structure and looking at joint chemical structure between the two is performing very well. It's performing better than some traditional methods and some methods which also include protein information. So in our case, we haven't looked at protein information, and that could be one further work on how to make this method probably even better. And here there's an ablation study. So how the individual parts, so is the cross-drug interaction useful or not? And we see that the different components of our architecture are helping. And we've also looked a bit at how the performance is on individual side effects. And we can see that so some of them are perform very well, but then some of them don't perform that well. And it might be that those which don't perform that well is because maybe they're not, there's no causation between them in a way. So for example, having the HIV disease as a side effect is probably something that was correlated and caught in the data, but not something that's caused by taking two drugs. And we also visualized the 200 learned drug representations across 9 side effects, and we see that the TSNE, which is a visualization method, clusters them in a representative way. Okay, so so far we've covered how to preempt side effects, which is, as I was saying, something that's done in phase 4 drug monitoring phase. Because it can't be done exhaustively before the drug is put on the market. But drug combinations are not only causing side effects, so they're not only interfering, they can also be helpful. And we can use similar tools, so graph neural networks in a way to find potent molecular cocktails. And one example is for patients who have had a heart attack. So after 1 year from the incident, they're usually taking a combination of drugs such as ACE inhibitor, beta blocker, aspirin, paracetamol, antidepressants. So all of these are usually administered together in such a case. And in order to look at positive drug combinations, so drug combinations is a way to treating something rather than having a side effect, as a new way of treating something. I'm going to first revisit what we were talking about. So we're going to use the chemical graph, where, as I was saying, the atoms are nodes and the bonds are edges. But now we're also looking at the target protein. So what we know is that a drug is designed for a bunch of proteins, which is going to target. So this is the group here, and this is the group for drug 2. And in this work, we looked at the interactions between the drugs. So GC represents the chemical graph, and P's are the protein targets. And we find different ways of modeling the interactions between the chemical structure and the protein, either through direct interaction or through the interaction of the chemical structure with a summary of the protein. And this analysis also showed us that there's ways to combine this information in such a way that it's better compared to taking the chemical information by itself or the protein information by itself. So a better model would in this case work if we combine this information, so the chemical and the protein together. Some conclusions from what we've been talking. So the graph representation learning has the potential to transform healthcare, and the molecules are naturally represented as graphs because, as we've seen, there's a simple mapping we can do and not discard some information. And then every stage of drug development pipeline is amenable to GNNs. And we also explored only the aspect of drug combinations. If you'd like to find out more, there's this archive link. Thank you. If you have any questions, feel free to email me or find me on Twitter. Yeah, this is it.
Very good. Thank you very much, Andreea. Very, very detailed and very much appreciated. Can I ask you a more macro level question, please, Andreea?
Of course.
Maybe you have some perspectives on what you've heard this morning yourself. Had you any reactions to the earlier conversations we've had in relation to research, clinical trials, etc.? Were you able to catch any of those talks?
I wasn't able to. If you can, just tell me a bit.
Just there, I seen a good overlap, and I wondered if you had a perspective on that. The question I had for you actually was in relation to different disciplines. Again, as a non-expert, my interpretation of what you've so thoroughly presented is scientific, that your outcomes remain the same, irrespective of how we choose to get there. In other words, how we may look at the drug applications for AI versus the same drug applications without AI. Have you a perspective on artificial intelligence and how it can be useful in these contexts, given the times we live in, and of course, the same universal pressures of faster innovation, faster modeling, etc., etc.?
Yeah, so there's I guess the bigger question is how AI can help drug development, like the whole pipeline. And there's a lot of ways in which it can help. But I guess the main point, the main aim is something that reduces cost or time, because drug development, the whole pipeline takes a lot of years, so around 10 years, and it takes tens, hundreds of millions to develop. But if we could use machine learning to, for example, shorten the time it takes to find an initial good molecule, so an initial good drug, by looking through millions of registered, already recorded molecules and their activities, that would already make the time frame shorter. Or so drug combinations is one such example. If we use it to predict that these two drugs will have a very bad interaction, then maybe we should be careful and we should not recommend people to take these two drugs together. So in this way, saving them the costs of having the side effects and then having to treat themselves or taking some additional things to feel better.
Yeah. Okay.
So, this is
very good.
examples.
Yeah, very helpful actually. And then the relationship of, if you like, seeking, how would you describe the funding, the related funding and applications? I'm quite sure, I dare say, that AI can be very useful if it can demonstrate its ability to shorten those timelines too. I have a couple of other questions, if I may. Alexander says, what's the size of your graphs and how big the dimensions and features in the nodes can go? How big can the dimension of the features in the nodes, how big can they go? Does that make sense?
Yeah. So the graphs are, I guess it depends on what we're comparing with. So the graphs are usually from tens of nodes, so somewhere from 20 to hundreds, but not like 100 maybe. So they are not very big. They're not nowhere near as big as transportation networks or that.
Tens of nodes, right?
Yeah.
Okay.
And how big the dimension of the features? So I think this depends from task to task. It can be something as wide as 32 or something wider, like 512. So I guess there's some. It depends on how complex and how much data you have. With more data, you can do more parameters in a way.
Okay. And Stojan asks, have you tried BioVia software?
I am not aware of this, but I'll take a note and look into this afterwards.
Okay. So that's a no. Thank you very much. That's a no. And then Alas asks, Here's the complex question, so bear with me. Given the state of AI in health, is perhaps some more general approaches to computational address the society and also include the optimization model, the model of having the drug used and confirmed in all phases, even phase 4, e.g. in phase 4, most possibly this would involve not only modeling human cells and organs and body status, but also current economy and administration. Okay.
Yeah, so I guess I don't know about economy and administration modeling. I think there's quite some way to go until we go from here to there. But there's more intermediate steps we can use. So as I was saying, we're already looking at drugs and proteins, modeling more complex things in the body, like the metabolic network and things like that could be useful. And sure, from there we can go to even larger scale things.
Okay, very good. Any more questions? I don't think so. I think that was pretty clear. So yes, have you any final remarks at this moment then, Andreea? Anything you'd like to add?
I encourage people to work on this. I think it's a very exciting direction with a lot of potential.
Yes.
It's been very, very exciting to work on this at this point. Maybe in a few years it will be way more developed. So I think it's a good train to catch.
A good train to catch. Well put. So on that note, we'll end it for now. Thank you very much, Andreea Deac, AI researcher at Mila and the University of Montreal, PhD candidate. Thank you very much. And of course, if anyone has any follow-up questions or more questions on the back of those statement questions, we can by all means contact you. Thanks very much, Andreea.
Thank you.